Welcome to Chapter 4, focusing on prokaryotic diversity. We're going to look at where prokaryotes live, how they interact with each other and with us, and how scientists classify them. Then we'll start looking at the major bacterial groups, starting with Proteobacteria. Chapter 4 is divided into 6 sections. We'll start with habitats and microbiomes, then look at Proteobacteria, non-Proteobacteria, gram-positive bacteria, deeply branching bacteria, and archaea. So in this video, we're going to cover the first 2 sections. By the end of section 4.1, you should be able to describe where prokaryotes live, the different types of symbiotic relationships, compare resident and transient microbiota, and explain how prokaryotes are classified. Prokaryotes are single-celled organisms without nuclei or membrane-bound organelles, and they're literally everywhere. They can be in the soil, the water, the air, and in extreme environments like hot springs, salt lakes, and deep in the ocean. They also really thrive in and on our bodies, especially in the mouth, skin, gut, nasal cavity, and even in the female vagina. They are metabolically flexible, and that means they can switch energy sources depending on what kind of nutrients are currently available. They're also essential for the balance of the ecosystem and the cycling of different nutrients like carbon and nitrogen. This image shows halophiles, which are salt-loving microbes, and over here on the left is the Dead Sea, where you can find such microbes. One example that you could see in there, or find in there, are Halobacterium salinarum. These prokaryotes can survive and even thrive in conditions where few other organisms can because it's so salty. So the Dead Sea is located in the Middle East. It's actually known as the lowest point on Earth, where it's about something like 430 meters below sea level. It's also one of the world's saltiest bodies of water. So super high salinity, has an average salt concentration of about 34%, which is nearly 10 times saltier than the average ocean. Prokaryotes perform vital ecosystem roles for us. They can fix carbon, for example, converting inorganic carbon dioxide into a usable form of organic carbon, such as glucose for us. They can fix nitrogen, such as rhizobium, the bacterium found in legume roots like beans, peas, or lentils. They can improve soil fertility, so they can break down organic matter, forming that organic layer within the soil that's called humus. They can clean up pollution through bioremediation by getting rid of pollutants and even toxic chemicals. Very few are actually pathogens, so fewer than 1% of prokaryotes are disease-causing, but of those few, they can cause many different types of human diseases. And they can also influence the climate. When permafrost thaws, microbes are released, and those microbes can release greenhouse gases like carbon dioxide and methane, which contribute to global warming. This figure shows that bacterium rhizobium living in the root nodules of different legumes, these different peas or lentils, and you might have seen them before if you've ever pulled up the roots of these plants. These bacteria convert atmospheric nitrogen into ammonia, which plants need to build biomolecules. Here's our checkpoint. In what kind of environments can you find prokaryotes? So pretty much everywhere on Earth, in the soil, in the water, even in the air, even way up high in the atmosphere. In extreme environments, we have the hot springs where you find microbes, the salt lakes like the Dead Sea, Antarctic ice, deep ocean pressure zones, and on and inside the human body, we have microbes in the skin, the mouth, the gut, the respiratory tract, the vagina. In man-made environments, microbes are capable of degrading pollutants and toxic chemicals. For the second one, name some ways that plants and animals rely on prokaryotes. We talked about carbon fixation, so microbes can convert carbon dioxide into organic carbon that animals can use. Nitrogen fixation, like rhizobium in the legume roots, can produce ammonia, which is essential for plants and their building of biomolecules. Soil fertility, we talked about how microbes can break down organic molecule to form humus, and that supports plant growth. And upcoming in the next few minutes, we're going to talk about mutualism in animals. So we have microbes in our gut, like E. coli, that can provide vitamins for us, such as vitamin K for blood clotting, and even a defensive role. So microbes include like lactobacillus, a good bacterium that can be found in the vagina that produces lactic acid and prevents yeast overgrowing. Many different types of symbiotic relationships exist between prokaryotes and humans. Symbiosis means a close interaction between species, and these are the different types of relationships. In mutualism, both species benefit. An example could be Bacteroides that live in our gut. These can convert plant carbohydrates that we cannot digest into a version that we can absorb. E. coli also lives in our GI tract and produces vitamin K, important for blood clotting. So we benefit from these microbes, but they also benefit from us because they get a safe, warm, moist, nutrient-filled environment when they live in our GI tract. In amensalism, one species is harmed while the other is unaffected. And an example could be Staphylococcus epidermidis and Cutibacterium acnes. These are normal skin bacteria that can release bacteriocins that are toxic to other bacteria and can kill them. So these normal skin bacteria are unaffected, but the bacteriocins killing these potential pathogens, these are the affected or harmed species. In commensalism, one benefits, the other species is unaffected. So Staphylococcus epidermidis can also be an example here, where they benefit from living on our skin and feeding on our dead skin cells, while we are neutral or unaffected. Although you could actually also classify these staph bacteria as a mutualistic relationship, because we also benefit sometimes as a side effect when they protect us from potentially harmful pathogens. So you can see many of these relationships can fall under more than one category. In the next neutralism, neither species is affected. An example is Bacillus anthracis that can be in its endospore state and undergo germination into normal vegetative cells again, and they can continue to exist with other endospores without harming one another. And finally, parasitism is when one species benefits but the other is harmed, and this is the case anytime a microbe is making us sick. For example, in tuberculosis or tetanus, these bacteria are benefiting from living and feeding off our nutrients, but we're being harmed because these bacteria are causing disease in our bodies. Our book provides a really nice summary table. I recommend kind of covering this part and seeing if you can define each one and provide examples for each one. Microbiota refers to microbial communities on or inside of our bodies. So these are all the different microbes that live on and inside of us. And there are two subcategories. Resident microbiota are basically the microbes that you've had on or inside of you your whole life since you were born. They're basically like your best friends. So these are stable, long-term colonizers that were transferred to you and basically at birth and have been on you for years. Transient microbiota are temporary. These are things that you pick up daily, like by touching a doorknob or eating certain foods. And these can also include disease-causing microbes, pathogens. Both your hygiene and your diet can alter resident and transient microbiota. So even residents that should be long-term colonizers can turn over. They can change. In contrast to microbiota, the term microbiome refers to all the microbes, the entire collection of microbes and their genes in a given environment. Different body sites will host different types of microbes. For example, in the cheek, we have Streptococcus that is dominant. In the tongue crypts, so the indentations in the tongue, we have anaerobic bacteria as well as many other types of microbes that are anaerobic, including Bacteroides. Bacteroides is also dominant in the intestines. Babies actually get their microbes first at birth, and the mode of delivery will affect their early colonizers, the types of microbes that will be on them, their resident microbes. And there is a difference in the mode of delivery. So the mode of delivery can affect colonization. There's a difference in the microbes of babies that are born vaginally versus through C-section. Your microbiome can also change constantly. For instance, in the small intestine, the microbiome can shift, can change within hours depending on your diet. And there have been a lot of studies telling us about how eating processed foods can change your gut microbiome, and that can influence the different factors that they release, the bacteria release, which can influence how you feel, right? These can travel to the brain and influence your mood. We can also have disruption to our microbiota through, most commonly, antibiotics. Antibiotics, when you're taking them to kill some kind of bacterium, they often kill even the good bacteria. So one of the most common examples is when females on antibiotics for something like a urinary tract infection develop a secondary infection like a yeast infection. So why this happens or can happen is because taking antibiotics can kill the normal vaginal bacterium Lactobacillus, which usually produces lactic acid to lower the pH of the vagina and prevent yeast from overgrowing. But when you kill off the lactobacillus, there's no more controlling the yeast. The yeast can overgrow and now cause a yeast infection. The same thing can be true for gut. If you're on antibiotics, that can kill your normal gut microbiota, causing others to overgrow, and that can cause other symptoms like diarrhea. And we'll talk about that further soon. So this link takes us to an older review article, but at the time it was really big when it came out. It talks about the diversity and colonization pattern of baby gut microbiota, so of newborns, and how they were significantly associated with the mode of delivery, if they were born through vaginal birth or C-section, especially during their first 3 months of life. There were fewer infections for the babies who were born through vaginal birth. But after 6 months, there was no real significant difference in the gut microbiota of either group. And that was probably due to breastfeeding and bottle feeding over time. But this is important because in those early months of life, the baby is still developing their immune system. So having fewer infections can be much safer for these newborns. And this led to current clinical trials where they're trying to see if babies born through C-section that get a smear of their mom's vaginal fluids will be healthier. So this is currently undergoing, so we'll see those results hopefully soon. All right, another checkpoint, cooperative versus competitive interactions. So I didn't talk about these in my previous slides because I thought they were kind of self-explanatory, but let's briefly look at these words. So cooperative means different populations are working together, benefiting from each other's presence, such as different microbes sharing nutrients with each other. Whereas in competitive interactions, it's what it sounds like. Populations are competing for some kind of limited resource. So one bacterium could release toxins that kills the other types of bacteria so that that original bacterium can have all those limited resources for themselves. In the second checkpoint question, we have list the types of symbiosis, explain each one. Go back and look at that chart. We talked about mutualism, where both populations benefit, amensalism, where one is harmed, the other's unaffected, commensalism, where one benefits, the other is unaffected, neutralism, where neither population's affected, and parasitism, where one benefits but the other is harmed. And make sure you can give some examples for each one. Taxonomy is the science of classifying and naming living organisms. And classifying prokaryotes is very tricky because they don't reproduce sexually and they lack a lot of distinct features. For example, most of them are already spherical or rod-shaped, so it's hard to tell them apart. So in traditional methods, we used shape, gram staining, and biochemical and physiological traits to classify them. And in modern methods, though, today, one of the best methods is through nucleotide sequencing. So we can sequence their DNA or RNA, such as ribosomal RNA. That's the gold standard. It works really well because you don't even have to grow the microbes in lab before you sequence them. Bergey's manual, which was first published in 1923, is still an important key reference, and its systematic bacteriology version and volumes are continually updated using approved names from the LPSN, which stands for List of Prokaryotic Names with Standing in Nomenclature. So this is a standard that all microbiologists follow to ensure that we're all using the same name for a microbe. An older version of Bergey's manual is now available to everyone online, and I wanted to point out, for example, Streptococcus salivarius. They say that these bacteria have spherical or ovoid cells that range from 0.8 to 1.0 micrometers in diameter. They're arranged in chains. These are gram-positive. Just to give you an example of what you can find out about a bacterium in Bergey's manual. Gram staining is still fundamental to classification of microbes. Remember that gram-positive bacteria have a thick peptidoglycan wall and stain purple, such as Bacillus anthracis. These are gram-positive bacteria. Remember that gram-negative bacteria have a really thin peptidoglycan wall and they stain pink or red after the gram stain, such as E. coli. We also have atypical bacteria like Mycoplasma and Chlamydia that don't fit the pattern. Bacteria that cannot be stained by the gram stain procedure are called atypical bacteria. Rickettsia are also considered atypical because they are too small to be evaluated by the gram stain. We have many subgroups of prokaryotes, and major subgroups include deeply branching bacteria, which are very ancient, early evolutionary forms of bacteria that live in extreme conditions, such as hot, acidic, UV-exposed, and anaerobic conditions without oxygen. We have Proteobacteria, which is also a very diverse group. This is a diverse group of gram-negative bacteria. The CFB group, we'll talk more about these later, include the gut microbiota, such as Bacteroides. We have spirochetes, which are spiral-shaped pathogens, such as Treponema pallidum, which causes the sexually transmitted infection syphilis. And we have our gram-positive bacteria. Gram-positive bacteria are divided into two different groups, low GC and high GC groups. And G and C refer to guanine and cytosine nucleotides in DNA. Remember from your previous courses, DNA includes A, T, G, and C nucleotides. A pairs with T with 2 hydrogen bonds, and G pairs with C via 3 hydrogen bonds. The more G's and C's you have in your DNA, the more stable your DNA. So low GC gram-positive bacteria have less than 50% G's and C's in their DNA, and these include Clostridium, Bacillus, and Listeria bacteria, whereas high GC include Mycobacterium and Corynebacterium, which have more than 50% GC content in their DNA. So how do scientists classify prokaryotes? Traditionally by shape, gram staining patterns, and their biochemical and physiological traits. But in modern methods, we really do sequencing. So sequencing of DNA or ribosomal RNA for much greater accuracy. And we use our reference tools like Bergey's Manual of Systematic Bacteriology to provide us with standard taxonomy and approved official names. The Human Microbiome Project launched in 2008 to map microbes in humans. And because 99% of microbes can't be cultured, right? They don't grow in the lab or on a petri dish. Researchers use metagenomics to analyze their DNA directly. And the project results were really interesting. So far, they've revealed that everyone carries some kind of pathogens in their normal microbiota, in their normal flora, raising questions about why some are harmless in some people. So why are some of these microbes harmless in some people, but pathogens or disease causing in others? The project also discovered many new species that were never grown before in the lab, so we didn't know they existed except or until we saw they existed through their DNA. It also aims to link microbiome composition to disease. So we want to find out whether there are any relationships between the contents of an individual's microbiota and our risk or susceptibility to specific diseases. And analyzing the microbiome in a person with a specific disease might reveal new ways to fight these diseases. Let's recap 4.1. So in the first question, identify and describe unique examples. We found that microbes are found everywhere, right? Prokaryotes are found in the soil, water, air, extreme conditions like hot springs, the Dead Sea, Antarctic ice, and even the deep ocean. We had halophiles that thrive in salty environments, such as Halobacterium salinarum, that archaean that lived in the Dead Sea. We had nitrogen-fixing bacteria like Rhizobium and legume roots that convert nitrogen gas into ammonia. We have soil bacteria that metabolize organic material, and bioremediation bacteria that can degrade toxic chemicals in polluted environments. For the second one, symbiotic relationships, we talked about mutualism, amensalism, commensalism, neutralism, and parasitism. So make sure you can define and give examples for each of those. Looking at normal commensal resident microbiota versus transient microbiota. For resident microbiota, these were long-term stable colonizers, such as lactobacillus and the vagina. Commensal or normal microbiota are microbes that normally live on or inside your body, usually harmless or even beneficial. We had transient microbiota, which were temporary residents, though they could include pathogens, and these were influenced by hygiene and diet. So some differences include that residents are consistent and protective, while transients really come and go and can sometimes be harmful. Looking at how prokaryotes are classified, we talked about traditional methods like shape, gram stain, and biochemical and physiological traits, but modern methods use sequencing of DNA or ribosomal RNA for more precise classification. We also use Berge's manual as a standardized taxonomy and we know they have approved bacterial names. All right, in our next section, 4.2, we'll cover Proteobacteria, and by the end of the section, you should be able to describe features of each class and give examples of important genera. Proteobacteria are a large, diverse group, specifically phylum, of gram-negative bacteria proposed by Carl Woese in 1987. So this was based on his findings of their DNA sequence similarity. These proteobacteria include both normal human microbiota as well as many pathogens, and they're divided into 5 classes: alpha, beta, gamma, delta, and epsilon. We'll conclude here for the first video. In the next video, we'll continue with the first class, alpha proteobacteria.